In this video, we are going to answer the question. So let's read the question together. At the start of a Meselson and stall experiment in generation 0, which is here, the generation 0, a single band of DNA with a density of 1.730 gram per cubic centimeter was found. This is the unit for density. After 4 generations, 2 bands were found. It's here, generation 1, 2, 3, and 4. After 4 generations, 2 bands were found. But the main band had a density of 1.700 gram per cubic centimeter. Imagine that you are just reading this question, but you have to draw this picture by yourself. I think the explanation of this is going back to the previous video, so you need to go back and watch it again if you didn't watch it, so you need to do it again. The first question is, explain why the density of the main band changed over 4 generations. So why the density of the main band changed over 4 generations. Here is the density. The first band, and in 0 generation, the density was 1.730 gram per cubic centimeter. And then after 4 generation, the density of band is no longer here, and it is located at the middle and in the top. Why has this happened? Pause the video and answer the question. Okay, I hope you have already thought completely about how to answer this question. It's very clear that if you go back to this drawing, you will find the answer. So why the density of the main band changed after 4 generations is clear, because every time there is a separation of the DNA strands, and in the first generation we will have only this type of the DNA strands, and then in second generation we will have this type. Considering all the explanations starting from first generation, by looking at this picture and by these DNA strands that are already drawn, you can answer this question. In the second generation, we only have 2 bands, which one band is in the middle, which is a mixture of nitrogen 15 and nitrogen 14. But in the first band is only including nitrogen 14. So here we go. If you want to answer, then you pause the video and look at the answer over here. Okay, let's have a look at the second question. After 1 generation, still only 1 DNA band appears, but the density has changed. So it is talking about the generation 1, that in the generation 1 you will see this pattern. Estimate the density of the band. What do you think is the density of this band in here? Which, if any, mechanisms of DNA replication are falsified by this result? And explain why the identified mechanisms are falsified. Pause the video and answer all the questions. Okay, estimate the density of the band. It's clear that the first band, which was mentioned already as a 0 generation, the density was 1.730. And then we had a middle band, and then from the second generation until the 4th generation, we had a top band, which has been already mentioned inside the question, that has 1.700. So we have a 1.730 and 1.700. So it's really easy to calculate the middle one just by simply get the average from these two. So the answer would be 1.715, the density of the middle band. The second question, which mechanisms of DNA replication are falsified? Of course, conservative replication is already falsified because in conservative replication, all the original bands should be remained. But here, you will see that the original one, which was here, it doesn't show up anymore in none of the generations started from generation 1, 2, 3, and 4. So you don't see any more the original band. So there is no conservative replication. There is no conserved DNA band. And the third question is, explain why the identified mechanisms are falsified. For conservative, as I just explained, for conservative replication to be the case, two bands should appear in all generations after generation 0. So, and the reason is, there is no conservative or conserved bands, especially the original one over here. So the new bands already form, which is not a part of that theory, which shows another theory that Meselson and Stahl mentioned later as the semi-conservative theory. Third question, describe the results after 2 generations and which mechanisms and explain identified mechanisms, if any, are falsified as a consequence. Pause the video and answer the question. Describe the results after 2 generations. Of course, the result after 2 generations here is only 2 bands. So that's the results after 2 generations. Which mechanisms and explain identified mechanisms if are falsified. So of course that the mechanism that can be explained over here is semi or semi-conservative mechanisms, and the falsified theory, of course, are the conservative and also the dispersive. Last question, describe and explain the result found by centrifugation, a mixture of DNA from generations 0 and 2. Here we go. The question is between generation 0. So if this is a generation 0, then you will have this band, and generation 2, then you will have this band. So if there is a centrifuge mixing generation 0 and 2, this means that we will have 3 bands. So the answer is 3 bands. Okay, we continue after Meselson and Stahl experiment. I hope you understood well, and if you have any question, you can ask me later. The next slide is talking about transcription and translation. We have already talked about these 2 items very much, and you know that what is the purpose. So, of course, the purpose of the transcription and translation is production of protein, and proteins carry many essential functions in cells. So that's the reason and the purpose of transcription and translation, which ends up with protein synthesis. If you want to learn more about this transcription and translation, there is a tutorial video, so you can pause the video and then go back to your PowerPoint file and then find this link and click the link and watch the video. Here is a summary of what is happening in transcription. Simply, there is a messenger RNA produced during the transcription, which means that the targeted DNA, which is going to become translate to protein, first of all, it needs to be transcribed, which is happening inside the nucleus. And then this mRNA messenger RNA or mRNA, after making a copy from the DNA target, it leaves the nucleus, leaves the cell nucleus, and immigrates or travels into cytoplasm, and will attach to a complex which includes the ribosomal parts, and then with a mechanism which is called protein translation, then the amino acid chain will be created, as you can see, and finally it ends with a polypeptide, or depending on the protein structure, it might have more than one polypeptide to become a protein. So if you have any question regarding transcription and translation, you have to go back and watch the videos regarding 2.6 PowerPoint slide and study there. And yet here again another slide regarding transcription. The DNA, separation of the DNA strands, production of the mRNA or messenger RNA by the help of the RNA polymerase. This is the process of transcription. And finally becoming an mRNA. In that case, the mRNA leaves the cellular nucleus and travels to cytoplasm. One thing that you should know here is that it's not only the mRNA which will be synthesized from the DNA, but also there are another two types of RNA which are really important in the translation process. They are also synthesized from the DNA, and they are transfer RNA or tRNA, which is a clover leaf shaped sequence that carries out the amino acids. If you remember, it had some structure, something like this, that it had the anticodon over here and the amino acid over here. So that's the tRNA, and also the ribosomal RNA, which is a component of the ribosome. It's a structural part of the ribosome. So not only the messenger RNA is synthesized from the DNA, but also another two types of the RNA, which are called transfer RNA and ribosomal RNA, also synthesized. Here we focus more on the function of the RNA polymerase in the production of the mRNA. If you look at over here, you will see that this is the RNA polymerase. It is multifunctional and more complicated than DNA polymerase, and the reason is only RNA polymerase can do multifunctions, including binding to the site of the DNA at the start of the gene. So that's the first function, that is recognizing the place that needs to be transcribed. That's first. The second function is that RNA polymerase separates DNA strands. So this is the second one. Pause the video and tell me, do you remember what was the enzyme for the DNA separation of the two strands? What was the name? Yes, you're right. For the DNA replication, if you remember, there was another enzyme, not just the DNA polymerase, but also there were several different types of enzyme. And the enzyme which was responsible for separation was helicase. But here, everything is done by the RNA polymerase. So RNA polymerase will bind, which means it does the recognition part or function, and then it separates the DNA, which does the function of the helicase in DNA replication, and then synthesizing the complementary RNA by making a copy. So here is the third function, synthesizing. So three different types of functions which has been done only by one type of polymerase, which is RNA polymerase. So to me, is like RNA polymerase is much more advanced than DNA polymerase because it is multifunctional and it does three different types of things at the same time, only by one enzyme. Okay, once the RNA sequence has been synthesized, which is here, then what happens? The complex that has been created over here and the separation of the DNA, which has been just separated in order to transcribe, should go back to the original situation. So which means that RNA polymerase will detach from the DNA molecule, so it goes out, and then RNA detaches from the DNA, which means this RNA will be detaching from the DNA. So separation of the RNA polymerase 1, detaching of the RNA 2, and 1, 2, and 3, the double helix reform. So which means that these two strands should go back and reform by attaching to each other again. So A to T, A to T, T to A, and again the hydrogen bonds will be reformed. Once DNA travels to cytoplasm, then that would be the start of translation by attaching to the ribosomal subunits. So there are two subunits, small subunit and large subunit. Small subunit is the place for attaching the mRNA, and the large subunit has the places for binding to tRNA. It has the tRNA binding sites. So once the mRNA is moving through the ribosome, the tRNA will come one after the other, and then the previous amino acids it has the link between the previous amino acids and the new amino acid will form, and the chain will form like this, and at the final stage that would be the last amino acid with the last tRNA without any amino acid, which has the information regarding to the separation of the complex, and then the chain will be released as a form of polypeptide.